My name is Christine Wandelt, and I'm a Regulatory Affairs Manager with BASF Plant Science, and I will introduce you to a quality trait and the risk assessment of a quality trait, which is altered starch composition in potato. So we'll provide some overview of the trait, describe it, describe the crop and potential uses to set the frame for the assessment, give some background information on food and feed safety assessment, and also touch on some environmental risk assessment aspects. So what are the products? There are actually several I would like to talk about. They are non-commercial, meaning they have gone through a certain stage. You have seen yesterday one on the list of Laura of the products about what have we learned. So this is the Amflora. The two others, they were developed to the point where applications were submitted to the authority but withdrawn at a later point of time, so they could also go on the list as Laura presented. And they all have names because they are in potato, and hence the event you select is also the final product, and they have gone through a process to register them as varieties. So this is why they are called Amflora, Amadea, and Modena. They have all their very specific history. Amflora has the longest history. If you look at the event name, it starts with the initial of the scientist, and then there's a year 92, so that's the time when it was developed. And at that point in time, there was the idea that, and I think that was also how it developed on a legislative point of view, that an event could be, or that a construct could be, would be the entity that is regulated. So if you would use the same construct and transfer it into different backgrounds, then you would be able to include it in one application and then obtain approval for maybe 3 different varieties that come out of it. So we quickly learned that this is not possible in the current system, and it's regulated on an event basis. Hence, they are all distinct events with distinct constructs they were transformed with. One without any selectable marker, which is the Modena. The 2 others contained selectable markers like the NPT2 gene in the case of Amflora, and the herbicide tolerance, what you know from clear field crops, what you have heard before, in the case of Amadea. The method that was used for transformation is the agrobacterium-mediated transformation. Important to know is also that this is a vegetatively propagated crop, hence you have no option to breed, and what was mentioned in previous talks before, to select against any unintended effects. Nevertheless, also potatoes go through selection processes in tissue culture and in the field, and you select the clones that have your optimal characteristics you would like to take further. Just some information on the trait. What is it all about? This altered starch composition, this increase of one component of the starch, which is the amylopectin, the branched form of the starch. If you heat a solution, a water solution with a starch, a native starch, like here, and then cool it down, in case you use just a conventional starch, it will gel, thicken gel. Whereas if you use an amylopectin starch, it will just thicken without gelling. Also, if, for example, you use oxidized starches, here a conventional potato starch in a solution, and heat it and cool it down, this is going to be turbid, whereas the amylopectin starch is staying very clear. And this is properties that are then really favored by in various areas. So it improves product qualities, also optimizes production processes, and leads to long shelf lives of products. So to just look at the safety assessment, how did we go about to develop our assessment, our data packages for all three products? Of course, there is the guidance by international frameworks and national legislation to focus on. Common for all is that what you are protecting is human and animal health and the environment. We applied a comparative approach, as was outlined several times already yesterday and today, in order to assess any identified differences relative to the conventional counterpart. I then also had a look and went back how are unintended effects described and what are you supposed to do in order to detect unintended effects, and Codex gives guidance on that. So it specifies where you can find them, where to look for. So it's described their inherent and general phenomenon that can also occur in conventional breedings. That's what Codex says. They are largely predictable based on the knowledge of the inserted trait, its metabolic connections, or of the site of insertion. You can identify them and detect them, and then evaluate their biological relevance and the potential impact on food safety. You should take into account economic and phenotypic characteristics that are typically observed by breeders as a first screen. So this is our starting point. Looking at the genetic modification, and if we find any effects that would point us to any harm, of course we start with what is the pathway about. This is depicted here. ADP glucose is in the amyloplast in the tuber, the substrate for starch synthesis, either soluble or granule-bound starch synthesis, and they take it and produce the amylose, which is the long-chain starch, unbranched, and then you have starch-branching enzymes that act on it at the branches, and you have the amylopectin, the branch-type starch. Usually, if you look at potato starch in the tuber, you have 80% amylopectin, 20% amylose. We applied an antisense. In the past, it was called antisense. We know now RNAi approach, in order to switch off the gene that leads to the synthesis of amylose. That's the granule-bound starch synthase, and therefore interrupted the pathway here. And the result was that there's only amylopectin that accumulates here in, for example, here the amphora starch. There is also a great data set on characterizing the insertion site in order to identify if there are any open reading frames that were newly formed or that were disrupted. However, it's, in the case of potato, not an easy undertaking, because the sequence, the genomic sequence that is known in all detail for crops as maize or soybean, is not really known for potato. Potato is a tetraploid, and so far it was only possible to sequence a diploid, a synthetic one actually, and it also shows us that there's a great genomic diversity in the case of potato, which makes it very difficult to sequence it. Nevertheless, you can also turn it around and can say if there's so much diversity, then if you introduce your gene at one location and you have three other genomes to compensate for, this might not make such a great effect. In order to show that our antisense approach, RNAi approach, works as intended, we can measure the inhibition of the enzyme, so looking for the granule-bound starch synthase, not only in the tuber but also in other tissues. We used a tuber-specific promoter to drive the RNAi construct, and we can also measure the metabolite, which is the amylose. And in all cases, it was confirmed that it's only the change that occurred in the tuber. Important, and as also outlined yesterday, it's relevant to set the context for your assessment and what is the intended uses, what are the exposure routes. And for those particular products, they were only intended for cultivation in the European Union, not elsewhere, because this is a product that is valued in the EU. Most of the starch in the EU comes from potato and hence has no value for other countries. Since it's a quality trade, you need to protect your trade in the field, hence set up an identity preservation system to grow it. It's not a commodity, so you do not want to have any mixing of a conventional potato with your amylopectin potato. Interesting is also that the genetic background that was selected for the transformation, the 3 different parental varieties, are so-called processing potatoes, which have a very high content in starch. So they are exclusively used to process them into starch. They are not used for producing table potatoes you buy in the supermarket, or the potatoes where you derive French fries from. The starch is used in paper, textiles, adhesives, but also in the food area. You have one byproduct of the processing, which is the pulp. It's just the fraction that's left over after we remove the starch, so it's high in fiber, high in protein. Important to point out, I believe, is familiarity. So how familiar are we actually already with this particular trade? What do we have to consider when we do our food and feed safety assessment? We know that amylose and amylopectin, they are components of all starchy foods. So we have it in our diet with whatever we eat, if it's the bread or the pasta or any other starchy food. The ratio of amylose to amylopectin, meaning the lack of amylose, is already very well known. You might have heard the term waxy starches. You know them from maize, but also from potato. An amylopectin starch, as I said, has a specific functionality, and usually this functionality is also put upon normal starch, native starch, via chemical modifications. A lot of starches you use for various applications, they are chemically modified in one way or another. Another point to know is that there is already cultivation of low amylose potato in the EU, because there are conventional amylopectin potatoes around, created by mutagenesis techniques and then very laborious breeding to arrive at that variety. It was mentioned yesterday as well that you might have effects of somal clonal variation, unintended effects. I said potato is propagated in tissue culture. So if somebody is concerned that via the transformation procedure you produce unintended effects to the product, you have to keep in mind that any potato variety you maintain and you propagate goes through an in vitro culture step till you then propagate it in the field. So you always maintain your stocks, you go into the field and you remove them again, and you look for off types, remove those off types to really have a clean stock. So let's move to the comparative assessment, and in particular the compositional analysis. We oriented ourselves towards the OECD guidance documents that for potatoes specify which anti-nutrients, nutrients, vitamins and minerals need to be analyzed. In particular, potato has one toxin, which is the glycoalkylate, so it's very important to not neglect that. We should focus on the pathway, in our case starch metabolic pathway, and possible changes to the metabolites, which are the sugars or related compounds. Sort of look at the magnitude of any effects, but also consider if we see any effects, what's the biological relevance of those effects. Here is, as an example, analytes we measured Those are the starch, the glucose, the fructose, the sucrose, vitamin C, and total glycoalkaloids. They are the three potato varieties, Amflora, Amadea, and Modena, and they are always compared to their parental variety and to reference varieties. And there's only one case where we saw that the sugars are elevated, statistically significantly elevated as compared to either the parental variety or the conventional reference varieties, but that was not for the other two. We also can see a trend with regard to an increase in vitamin C versus the conventional ones. There's also a trend where the glycoalkaloids are decreased versus the conventional ones. However, all those effects sort of disappear against the variability that is imposed by the environment and by the genetic background in the case of potato. This is also illustrated by a chart taken from a publication where in the Netherlands they looked at one particular potato variety, Ehud potato, that was grown year after year starting in 1978. Data were collected on the content of protein, of sugars, and of starch, and up to 1993, and you see how much it varies, especially for the sugars, depending on where you grow it, at which location, and what the climate was in that particular year. So all those components we analyze in the compositional analysis, they have high variability, which is maybe not unexpected because the potato as such has to adapt to the climate, to react to the climate, has to react to the environmental stresses. A plant can't not move away, so they have to keep their plasticity with that regard. This is another example where we look at the environmental influences for one particular variety and three reference varieties at 5 different locations, 1, 2, 3, 5, and 4 different minerals. The tuber is grown in the soil, hence you have uptake of minerals, and we wanted to understand if there is the effect between those varieties is larger than that comes from the location. And there's for the different minerals the reaction is very different. You might have a situation where the varieties are all very close and the location doesn't have such a significant effect. For sodium, for example, you have a dramatic difference with regard to the locations, but also that one variety reacts very differently versus the other varieties to the environmental influences at this location. Let's move to the economic and phenotypic parameters as a screen for unintended effects. Typically in potato breeding, there are a certain set of parameters that are measured. Amongst them is plant height, tuber yield, tuber starch content, growth habit, flower frequency, fruit frequency, the plant development parameters like days to emergence, crop vigor, plant stand, uniformity, the days to flowering, but also the reaction of the plant to diseases and pests and the severity of infection to virus, to Phytophthora infestans, to Avinia alternaria, or the degree of infestation with Colorado potato beetle. If you look at the ratings, just to mention that we use the same parameters as breeders use to assess new varieties, and there is a catalog you can look at by the the International Union for the Protection of New Plant Varieties. There is quite some confidence in the ratings because skilled breeders really can distinguish varieties based on those criteria and then sort of give them a variety registration but also keep them pure. You can also check as reference their catalogs published. When we looked at one This is the Modena versus the mother variety and the conventional varieties. For a set of those agronomic phenotypic characters, they were almost indistinguishable. It was to the point that if it weren't for our amylopectin trait, this variety would not have been recognized as new varieties. So they're very, very similar. We also, of course, need to look at what would be the effect of cultivating that crop in the environment, and provided by the EU, there's guidance as to which risk areas to look at. And so it's persistence and invasiveness, interactions with target organisms, but also with non-target organisms, impacts of management practices, and effects of biogeochemical processes. With regard to persistence, potato is a crop where tubers might be left in the ground after harvest, and the next year you have volunteers emerging depending on the environmental conditions during winter. So if you have very harsh winters, then the tubers will die and they will not emerge. And so it's very important to know if the trait, the changed starch composition, would in any way impact this characteristic in the soil, that volunteers can emerge. And we developed some tests to subject the tubers to various temperature regimes at various depths in the soil in order to understand if there's any impact on the ability of the plant after having been exposed to those temperatures to really germinate. And when we compared it to a set of conventional varieties, there was no real difference. There's great variability, of course, and no difference in the ability to have more or less volunteers. Invasiveness, including gene flow, is not a topic for potato because for one, most potato varieties, conventional ones, are male sterile, or they produce very little pollen, or the pollen is very heavy, only transported short distances by bumblebees. Interactions with target organisms, it's not a topic because we do not have a trait that has a target organism. We looked at interactions with non-target organisms. The plant grows in the soil, so you have soil microorganisms, fungal communities there, and it was especially important for the Dutch government to ultimately understand, and they set up a program to fund studies under this ERGO program, ecology regarding genetically modified organisms, where the unintended effects could be if crops are being cultivated. And Modena was one of the crops included in those studies. And what they were able to observe, where there were some differences, of course, when you compare the potato to the conventional counterpart, but they are minor when considered in the context of the variability exhibited by different conventional cultivars, meaning the genetic background, and then the magnitude of the environmental influences, like the location, the climate, or the presence or abundance of non-target organisms. So there was a whole set of studies and respective publications that came out of this ERGO program, and all showing that the effects by the genetic background are more pronounced than any effect you might have by inserting a gene into your plant. This leads to the concluding remarks and the assessment for unintended effects of a quality trait. In general, the safety assessment process is the same as for any other trait, so you do not make any differences with regard to a quality trait. You focus on what is to be protected, provided by the relevant legislation. Of course, you need to understand what is harmful to focus your safety assessment. And the risk assessment is guided by hypotheses related to the crop and the trait, and how the trait was achieved, like in this case, via a change in the metabolic pathway and by switching off a gene. And you should apply a targeted approach. So, of course, important the use of weight of evidence approach and to consider familiarity. And I think it would be an excellent outcome of that meeting if we sort of could close the loop for Amflora, and if there would be the opportunity in future for some vegetatively propagated crops or for some crops that have traits we are very familiar with, to sort of regulate them via constructs, assess them via constructs, and not each individual event. Thank you.